move on to the discussion part, and then I think it's Anton will take over as the moderator for the discussion.
Great. Yes. Thank you very much, everyone, all the speakers. And yeah, we now invite all the speakers of the day for the discussion. Well, so let's get started. There were some great questions from the audience today. I think maybe we can start with one of them, which was the following. Let's say we have the model or models that can predict every single spike and every feature of LFP and other metrics. So very precise, very detailed model that can really replicate the activity in the brain very well. What would we have learned about the brain from a model like that? Would we learn some knowledge? Yeah, so please go ahead, speak up and let us know your opinions, what these models are good for.
Yeah, maybe I can, I think this is a really good question, and a question that is often raised. And I think, for one thing, I think it's one thing that Upi mentioned actually in his talk, why you need these detailed models. There's certain things, if you want to, what happens is understand what happens when you add drugs, for example, or if you have genetic variations in which affects the ion channels, things which are at that level, you can't get away with simplified models. But then it would be sort of like this black box model in some sense, where you put in drugs or whatever, there's different kinds of genetic variations and be able to predict it. But the second thing is that if you have this ideal model, this model that can do all these things, this is like the ideal test animal. You can turn on and off all parameters in the whole circuit, right? So this would be this fantastic starting point for taking the model apart and see what makes it work, what kind of problems or what, and so on. So I think we'll also get, this could be a starting point for making these more principled models, which are more easy to understand, for example, in terms of population firing rates and so on.
Other thoughts?
Also, sorry. It can also help to understand the measurements. You know, we have many different types of measurements that everybody is using, and many times people are a little too easy in terms of assigning measurements to parameters. But we just need to keep in mind that each one of these measurement modalities comes with its own measurement theory, and that's its own challenge, try to understand what exactly is it that we're sensing or manipulating.
Oh, this is actually a good point, that if we if you want to compute things like LFPs and actually spikes and the measurements, if you want to model the measurements, which I think you need to do, then often you need detailed models to sort of to make sure that you understand what you're measuring and what it tells you about the model.
Upi, you have your hands up.
Yeah, I mean, this is a great question. It's frequently asked, and one of the answers that I think is nice is that the process of getting to a model of this level of completeness and accuracy is what will teach you what's going on. You can't get to that kind of completeness unless you have understood what's happening both at a more abstract level, as well as having a lot of insights about what's going on in detail. And I think that's what the fun part of doing the modeling is. You get the insights along the way.
Thank you. Sean, Keith, Chris, you have some thoughts?
Yeah, so I guess I want to just double down on what Gata said, that to me, as I raised, if you're starting to be interested in more complex systems, they're almost impossible to disentangle when you can only record from a few neurons at a time or make 1 or 2 manipulations at a time. And if you have a model that you trust because it's accurate, and in this case we're sort of omniscient and we know it's correct, then you can go in and make subtle and nuanced manipulations to a host of things that are very specific and kind of guide you to a more direct answer. So I see it as an indispensable experimental tool. So the better the model gets and the closer it gets to reality, the more effective it becomes in driving our experimental progress.
I'll add something from the kind of from the experimental side, right? When for the data sets that we collect, there's a lot of different things that one could analyze, right? And a lot of different directions that you could go with it. And sometimes having models with specific predictions, even if the predictions turn out to be not correct, right, can actually guide interesting directions of analysis and really focus that down to like what the kind of critical questions are, as opposed to endless kind of exploratory data analysis and so on. So I think to us, it's always been very helpful to have models that you could use in such a way, whether they be detailed or not, to help us guide analysis of very large data sets.
Yeah, so I agree with everything that's been said up to this point in terms of the utility of such a detailed model, particularly for mechanistic understanding. Ultimately, though, you know, we need to link mechanism and the detailed biophysics and biorealism to theory and to computation and algorithm, right? So it would be unsatisfying if we could predict the precise firing of each neuron in the brain, but not understand what it was for, how it was involved in implementing a particular computation. And so there's that component, right? That I would add here. So it's a big part of the study, story, but it's only half in some sense. And I guess the final thing I would add, and this echoes what Gaute said at the start, in terms of once you want to start fixing brains, clinical treatments, it's really important to have such a detailed model to understand the impact of drugs and manipulations for controlling brain dynamics.
Anna, yes, please.
There is also a need to bridge between scales, you know. So just this morning, I guess afternoon here in Boston, we heard one talk about intracellular cascades and calcium homeostasis and phosphatases and all this kind of intracellular machinery. And then a few talks about this type of neuronal networks that never get to that level. But of course they do, if you think about this, because this is where all the, you know, pharmaceuticals are going to be acting as well. So if we want to be a little more practical here in terms of trying to, you know, find new treatments for mental disease, we kind of need to bridge the scales, and we probably need to have some sort of nested model so that we can go from one level to the other. We can look at this, how it's going to be looking to us through the view of non-invasive imaging, and then go to the scale of cellular networks and try to understand what's going on the level of populations of neurons, and then go kind of deeper down to the level of single molecules and all the intracellular machinery where most of the targets may actually be for the drug.
Great, yeah, thank you everyone. Those are all very good points. I just want to chime in. I think this question of understanding is often asked, right? You know, you have this potentially huge and very accurate model, and does it really add to the understanding? I agree with everything that you all just said. And just to elaborate a little bit on Sean's point regarding theory, I think that's very important. And these models, these very detailed models by themselves, they do not maybe provide the understanding per se, but they can serve as a testing platform, right? You can test your hypotheses and your theories under realistic constraints, and that should help guide and move our understanding forward. So there are some comments here from the audience. So Ted says, Upi, yes, the process of building the model is probably the most informative thing. And then Jason Moore agreed, you got to the top of Mount Everest. What does that get you? Getting there is the entire point. Okay, thanks. So there are a couple more questions here that I think are related, so maybe we can discuss them together. One is from Terry Sinowski. Actually, to me, he's asking, are you coordinating your data and model with the European Union Human Brain Project? So the immediate answer is, well, not directly. We have some conversations and collaborations, like with Gaute, for example, or with the Blue Brain lab, where we are working together on the standards. But I think this is an interesting point. I'm curious about the panelists' thoughts regarding, what are the best ideas, the best ways to do, collaborative development of such models across the continents. And a related question from Pankaj Gupta: are there ways in which these big questions be answered by crowdsourcing? Is IBL ideal for that, or there are shortcomings there too? So.
Well, maybe I can add this thing with the Human Brain Project, because this, I think actually when this paper came out through your group, Anton, actually one of the leading person in Brussels, I think, was sort of noting this and asking actually, I'm not exact, but it's quite high up in this EU system, asking, is Human Brain Project involved in that? And then I was actually forwarded this, and I said, well, actually we are collaborating, and I think now we're making in the Human Brain Project this e-brains infrastructure. The whole idea is that you can that people all over Europe, and I hope all over the world, should be able to run these like large-scale models on like supercomputers around Europe, so that you can actually, the neuroscientists, computational neuroscientists everywhere, can actually be able to run these models. And I certainly think that we should also get this model installed on this eBrains infrastructure, so that people can run it. So this, to make it accessible, just like we should with other models, like the one that Mikael Reimann will talk about for the Blue Brain and Human Brain Project models for the rat neocortex.
Yeah, Upi, please.
I mean, something which I think many people have said is that you need to have standards, you need to have open source if you want to be able to share models. And the INCF, International Neuroinformatics Coordination Facility, many of the other organizations, but that in particular has been pushing for these for precisely this reason. You want to be able to join efforts and reuse models and basically scale the kinds of work people can do by cooperation. And the more that we standardize and exchange, the better.
Other thoughts? Maybe some thoughts about the mode of organizing it, like there is the International Brain Lab. Is this the ideal approach? There are some shortcomings or things that can be done better or different?
Well, there is this, maybe I can just, this open source brain that is organized by, I guess, Porik Gleason and others. So where actually models are not, it's not just a place to upload like large, say, network models. It's also a place where models are sort of like developed and so on. So it's like more like this growth environment for a model. So I think that's an interesting idea, because these models are not sort of like a model that is and one done once and for all. And then, so it shouldn't be a static model in that sense. It should be more like a dynamic model where you can sort of develop it in the community.
Okay, thanks. So let me bring maybe another question here. So I think, you know, one big topic sort of throughout this discussion today, and I expect we'll touch upon that over and over again in the remaining 2 days, is about reproducing certain phenomena, right? And we are talking about multi-purpose models that should reproduce multiple phenomena. But can we talk now in this panel and raise some opinions, have a discussion about what are the key phenomena or brain signals that a model should try to emulate? Maybe some kind of priority list we can discuss. So is that something that is, you know, just background activity at rest, or is it stimulus evoked? Should we think about an individual animal and reproducing activity in an individual animal versus an average animal? Individual trial or trial averaged? Should we think about dependence on the brain states, awake behaving during sleep? So yeah, obviously all of those are important, but please express opinions about what would be your preferred ones, some priorities here.
I'll give a quick response, which I think it's really important to recapitulate single trial activity, right? So that's what the brain does. That's how animals behave and think and respond on single trials, right? And so if we want to capture the functions of the brain, which I think is one of the main things that we would like to understand, then single trial dynamics are really important to capture. And then of course, all of the other things you mentioned, neuromodulation, brain states will come into this, right?
Thank you, Sean. Other thoughts?
I would say for the question of states and behavior and stimulus and so on, it might depend heavily on which brain area one's looking at, but a lot of the areas that we look at, you know, I think would be not so informative to look at without the context of behavior, right? And I think obviously each behavior can have its own different feature to it, but I do think that's starts to engage the networks and states that are more similar to the states where they're actually used. But ultimately, I think considering state as a variable that one thinks about, whether it be related to neuromodulation or tasks or whatever, I think is something that would be quite exciting to build into models, and is sometimes not always an emphasis, but could be quite important, I think. And it was, you know, like I thought it was quite exciting from your talk, Anton, where you mentioned like with different visual stimulus, right, with different contrasts, right, you can see different effects in some of the simulations, right? So exploring those parameters, whether it's through the visual stimulus or neuromodulation or a different task somehow, or a state or behavioral state, I think would be an exciting axis to play with in models.
Actually, I'm thinking, I mean, what is the lowest hanging fruit in terms of understanding anything in cortex, right? I mean, if you look back at the development of quantum physics, which is a very maybe not the best analogy, but at least in the beginning, they focus a lot on understanding the simplest atom, the hydrogen atom. And when they did that in like 1925 or something, it was quite easy to generalize to other atoms. So I think, so like, what is the simplest? I guess it's, I think mouse V1 is maybe some sense the simplest cortex, or meaning that it's simplest in the sense that it's like the most, where we have the most data. So I guess to start with, sort of like what's the kind of the simplest situation where you can understand what the mouse visual cortex, in the sense that you can have like this, starting with your model, Anton, and actually make it agree quite well with what you see experimentally.
Thank you. Any other thoughts, Anna?
You know, I would try to embrace a little more complexity. When we were doing experiments in many years under anesthesia, we could use this very kind of reductionist approach where everything was trial averaged and we could really control individual parameters. And then when we switched to awake animals, in the beginning, we kind of tried to continue the same approach until we realized that we're doing something super silly. And this is that, you know, I think all of us agree at this workshop that most of the information is going to be in the ongoing spontaneous neuronal activity. And this spontaneous neuronal activity, of course, is only spontaneous because we're not causing it, but for the brain it's not spontaneous. So for the brain, this is the neuronal activity. It's just not caused by the experimenter. And so now, as long as we kind of ignore it and just try to use an awake mouse in a way that we used to do it under anesthesia, this is, I believe, just a bit of waste of time. Because even if we average out all the spontaneous activity, we get to look at neuronal activity that does not exist. And so when we get to kind of embrace the complexity and look at neuronal activity that does exist, it's not even clear to me to what extent it can be isolated to one single brain area. And this has to do with all this kind of large scale imaging where the Brain Initiative recently moved. And you see these pictures of global brain activity and you realize that the entire brain is working for every single task. And so the question really, in my mind, is what are the measurements that are available out there that the models can operate on? And as the measurements move to the large scale, it seems natural to me that the models also are going to be going large scale, just because in a way we're fooling ourselves by restricting our view to a single brain area, because that's not how brain normally works.
Upi?
Yeah, I thought I would just say something to annoy everybody in this group, which is that the simplest mouse might be a fly or a hydra or a planaria or something.
That sounds good to me. Not annoyed at all.
But you know, people have done this. There is a history of this in computational neuroscience. People have done this and tried to look at what was considered simple neuronal circuits just to realize that they're not simple. So there aren't any neuronal circuits that are simple. It's just a thing that does not exist, you know. People like Eve Marder that went and they tried to look at those animals that are supposed to do simpler things. Well, there's lots and lots of complexity there too, you know. So yeah, it's not clear to me how that's going to be helpful.
Yeah, I think there is a related question here from audience, you know, speaking of complexity. So from Justin, can we use neuronal models to imply the role non-neuronal cell types play in brain function? So anyone wants to comment on that?
So maybe I should comment on this because we'll do it a lot, you know. So in our world, we try to think about these neurovascular units that are little microcosms that have all the cell types in interplay, which is the astrocytes and microglia and anything else, vascular cells and everything which is out there. But there is still this fundamental idea that there are neurons that do computation and everything else is playing supportive role. So in our field, there has been some attempt to kind of turn things upside down and said like, oh, no, we really compute by like blood vessels, or the blood vessels by their activity constrain neurons. But there's no real evidence for this. If you look even in development, or if you look in developed brain under all those conditions, there is very clear indication that the system is built such that neurons go and do their thing. So it's true that all those other elements provide nutritious support and all this other support, and they're important in a sense that if you go to disease conditions, well, sure, all those different elements can break and you get all those different disease. But if you're just thinking about how neurons compute, I kind of believe that we can just focus on neurons and not on, astrocytes. I think that astrocytes would be important to support things such as plasticity. So sure, so they need, neurons, if you grow them on a plate, they don't grow very well. If you put astrocytes together with neurons, every neuron develops a big smile because they like to be together with other supportive cells. And this just kind of comes to show you that there is a very, very large and complex supportive system, but the fundamental computation is still done by neurons, my view.
Thanks, Anna. There is another question. We have a little time left, so maybe let's try to discuss one or two more questions. Here's one from Stefan. While discussing models with perfect fits, it's theoretically interesting. Most models will not be perfect. Question, when do you believe a model is good enough, interesting enough to study as discussed before? So I think it's interesting to hear a perspective on this from both modelers and experimentalists. Keith?
Oh. So, I mean, to me, a model is good when it makes a prediction about the intact biological system that is then borne out. And if it doesn't, then I feel like we have to go back and update the model to try to more accurately predict what the biological system is going to do. So I think that sort of closed loop interface, sort of recursive feedback between experiment and model is essential. And I guess that represents my position as a bench scientist at the end of the day.
Maybe I can sort of say something there, because I think it's a question of sort of what is, if you sort of want to compare two deep networks, this convolutional neural network trained for image recognition. I mean, they have this natural variability, meaning that the weights will not be identical, even though they function the same way. So I would say that if you sort of have, if you're able to sort of like, in some sense, if your model deviates less than the natural variability, it's sort of like a Turing test. If you show me the model data and you can't distinguish it from like the data recorded from a cohort of mice, right? Then I would say you're in like as good as you can get. I mean, we're far away from getting there. But I mean, it's something about what is the natural variability. It's clear that, and even if you look at two neuron models which you fit with conductances, if you start asking questions like, well, where really, I mean, on the micrometer precision of where these ion channels are, then they are not identical either. So I think this about what does it mean to be equal, or what does it mean to be close, is actually quite a deep question, and we have to think about like in each problem separately.
You know, in my view, the experimental and computational neuroscience, at least, are not standalone fields. So those of us that do experimentation, there may be a few hundred years ago, you could just ask a simple question, yes or no, and get an answer. And so here's your experimental study. But today we cannot do that, because just the complexity of our tools and our technical ability to acquire information is such that we end up with this huge amount of information where we don't have yes or no answers anymore. So now if you need to make some sense out of your data, well, you use some computational tools. So how much math you put into this can vary, but all of them, you know, are some kind of models. So maybe some of them are baby models and some of them are more complex models, but there's almost no more this experimental studies that don't require any kind of quantitation that needs some sort of framework where you need to put few equations together to understand what's going on, just to capture your data.
Okay, thanks. Any other thoughts here? Well, let's take one more question here from Elba Serrano. Can you discuss concerns or standards for data quality? The model is only as sound as the quality and rigor of data collection. So yeah, are we now in the time where the data in the field being collected are such high quality that they can be used for models a lot? What are the concerns? What are the recommendations to improve the situation?
I don't know if I have an answer to that directly, but I do think every data set that's collected has limitations to it and caveats to the measurement methods and so on. And I think it's important that people building models are aware of those, right? So that there's a nice connect between what are the strong features of the data and which are the more assumptions or caveats that come with each collection method. So I think, I don't know if there's like a universal standard that can be said for data quality, but I think having a nice connect between what's strong in the data and what the modelers choose to use from it would be nice, I think.
Keith?
Actually, sort of on Chris's point there, I think that you can kind of invert that and suggest that maybe a universal standard in, say, a model quality could be dangerous, because it would then, I guess, eliminate the ability to have variation in conditions and experimental approaches and things that do bear on the data output. And so you'd sort of be imposing a structured view through your model on what you might expect in a biological system. And so I think that trying to sort of weave together complicated experimental setups and paradigms and strains and training with models just takes hard work on a case-by-case basis, and it requires strict controls and being blinded and kind of all the things that we learn early on in science classes. I think that the idea is great of a international standard, but I don't think it's feasible. Sorry, I'm being pretty cynical right now.
No, I totally agree with Keith. You know, there's always going to be frontier questions which can't easily be measured, you know, at any given level of the techniques. And so you're always going to be working with imperfect data. And, you know, in cases where you have excellent data, well, it's nice to be able to make models of that. But often the fun part is where you don't.
I think that we need to give exposure to modeling students to actual experimental setups and vice versa. I think that this can go a long, long way, because it is super important, I agree, to understand the limitations. But it's very hard to describe it without ever seeing how this data actually are collected, you know? So if our neurocomputation students actually, if neurocomputational summer schools and courses out there just make an effort to maybe take advantage of experimental labs next door and just make sure that the students in the same setting get some exposure, so that's going to be maybe very helpful.
Yeah,
In our computational summer school, we dissected a squid. And there was this bunch of engineers and mathematicians and computer scientists they had an absolutely wonderful time playing around with the squishy sea creatures. So yeah, it really works.
Sean or Gaute, do you want to add something?
I wholeheartedly agree with everything that has been said here, primarily around understanding the limitations of each data set. I would say on balance, the data is getting better. And it's getting better because it's more well described. There's more emphasis around reproducibility, standardization, describing methods, sharing data. And so I have hope that, you know, in terms of like quality control, things are getting better. But ultimately, there are limitations of even, you know, if an experiment is performed to the highest standard, that particular modality, OPHYS, optical physiology versus electrophysiology, they have their own strengths and weaknesses, and one needs to consider all these things. And I think it's really important for experimentalists to be very open about this.
Yeah, great. And maybe I'll just add for myself also that for model building, one aspect is really important, which is systematic surveys, right? You know, for certain aspects, trying to understand the system, it might be sufficient to do an experiment where you characterize a particular cell type. But if you build a model with multiple cell types, you need to know that aspect for all of them. And so that's where you know, big survey type of research comes in. It's not the only one that should be done, but it's really important for modeling.
I also think if I could sort of just, I mean, it's obviously this thing of getting for computational neuroscientists to get experience in and see what's going on and how these measurements are done. But actually, I think also, actually, since our group has done a lot of work computing LFPs and so on, you actually learn quite a bit about the signal just from modeling it, like biophysically, that the LFP is a much more complicated signal, even in this ideal model world, the biophysically detailed modeling, that and then well, and often I would say, like, even experimentalists who measure this, they often have a naive idea about the interpretation of it, that there's so actually I think also modeling the signal, like doing the measurement physics, also this has a strong pedagogical component.
Okay, well, thank you everyone. Keith, yeah.
Quick question. So I've noticed that there are a bunch of questions that have come through in the Q&A that we haven't had time to get to, that might be more directed. Is there a platform for us to sort of subsequently answer those and post them for the attendees?
Yes, so we are going to save all of the questions that are currently in Q&A that weren't answered. We're going to send them to speakers who they apply to, and then we will include those answers in the transcript when we send those out after the workshop.
Perfect.
Yeah, excellent, thanks. Yeah, so we have to finish now. This is a very nice discussion. Unfortunately, there is a whole bunch of really exciting questions here left that we don't have time to discuss, but let's answer them in writing. Maybe I'll just leave everyone here with this question from Terry Sinowski, because this is, I think, really, you know, very important for the whole workshop. We have come a long way from the single neuron era. The distributed representation data are exciting, but is there a new conceptual framework? So yeah, what kind of new conceptual frameworks can we extract from all this data and models and their interplay? I think we all need to think about that. Okay, so thank you all. It was a great session today, and we'll resume tomorrow, 8 a.m. Pacific time. Thanks a lot.
Great. Thanks, everyone.